Welcome, everybody. I'd love to introduce Junhao Wen from Columbia University and Penn, who's been doing a lot of cool work on applying machine learning to, well, imaging and trying to, and medical applications. Yeah. Welcome, Junhao.
Thank you, Greg, for the invitation. So I'm Junhao Wen, and I go by Hao. And right now I'm the PI of labs at Columbia Radiology and Biomedical Engineering. And I also hold affiliation at New York Genome Center and also visiting assistant professor at Penn Radiology. So for today's talk, I'm just going to go through my overall scientific interest by developing and applying AI and machine learning in biomedical research. Specifically, I want to go through my research trajectory from imaging data like brain MRI to genetics and to other organ imaging like heart and eye, OCT, etc. So for today's talk, the topic is investigating human aging and disease at scale via AI machine learning, imaging genetics, and beyond the brain. So my lab at Columbia, I just want to introduce my overall scientific interest. So I mean, I was trained as a computer scientist, and I truly believe that AI and machine learning can see what we don't see as a human being, especially in the case of biomedical research. With that being said, I'm not claiming at all the AI machine learning method can entirely replace the critical role of clinicians like neurologists for brain disorder like Alzheimer's disease or radiologists, etc. And several keywords of my research. The first one is multi-omics. Like I mentioned earlier, for my I was trained as a computer scientist. For my PhD work, which I will discuss later, I have been focusing on mainly brain MRI for the brain disorder like Alzheimer's disease and frontal lobe degeneration, etc. And towards my independence during my postdoc and right now as an independent PI, I have been very interested in bringing additional omics data into my research. This includes genetics, genomics data, proteomics, and maybe in the future look at all those omics data at a single cell resolution. And of course, the core part of the research here is to develop and also apply those cutting-edge AI methods in different clinical applications. For example, classification of Alzheimer's disease patient at an individual level, or deriving what we call brain age using brain MRI data to quantify individualized deviation from normative aging trajectory. And of course, we have been also working on clustering methods to find those disease subtypes or dimensions. Hopefully, those tasks can benefit downstream clinical application like drug development, patient stratification for future clinical trials, and drug repurposing opportunities. And finally, another keyword here is multi-organ. Like I mentioned, with more and more evidence from our team and also from other teams in the research field, we have observed even let's say Alzheimer's disease has been considered as a brain disorder, same as disorder, but we know, for example, retina features like IOCT, they have been using to diagnose Alzheimer's disease even before the disease onset. That's a whole idea or the main motivation for our multi-organ modeling in brain disorder like Alzheimer's disease. So I'm gonna briefly go through I think 4 projects throughout my career so far. The first project is from my PhD thesis work, which I have been focusing on reproducibility in the specific context using AI and machine learning methods to classify Alzheimer's disease patient at an individual level. I think at that time, we know, I mean, this is a very well-known issue, reproducibility crisis, in modern research or scientific fields, far beyond biomedicine. And at that time, I think Nature performed a survey in the research community asking why the machine learning field reproducibility crisis in science. Of course, like I mentioned, what we normally call data leakage, which I will showcase in the later slides, it's a serious issue to oversell or to provide overestimated results in biomedical research. So to bring this to a very focused project here, we use AI and the machine learning methods to classify Alzheimer's disease patients using brain MRI data, specifically from 3 data sets, which is publicly available in the Alzheimer's disease AD field, like ADNI, OASIS, and ABLE. I think even and nowadays, this free public data has been the major like data forces like to study Alzheimer's disease. So in this paper, first we performed a systematic review, looking at previous literature. What is the reliable accuracy that one can use machine learning to predict Alzheimer's disease and at an individual level? On the table here, the upper table showed basically the surveyed studies which showed very well. Sorry, the upper table showed the study which showed consider like reasonable accuracy. For example, to classify Alzheimer's disease and health control, basically the best results they reported is like 90% accuracy. But the table on the lower part is a paper who report very optimistic results, nearly 100% accuracy. At that time, as a PhD student, I had this question to myself, like, if those study has performed so well, what should I do to improve the accuracy at that time? And dig into deeper, like into the details of those studies, we found actually that the study listed in the lower part of the table has what we call data leakage issue, which I will show later. And this highlights why, like, the importance, at least for the AD research field, that interdisciplinary expertise, our teams should be like formed to study this kind of experiments. And to showcase the issue, what we call data leakage here, first we want to benchmark the reliable classification accuracy using like 3 data set I mentioned earlier. First step is to perform rigorous or nested cross-validation, which will unbiasedly report the classification accuracy performance on the on-film data, test data. So to do that, we first perform the training and validation test for the nested cross-validation. That means we also have a cross-validated test data set to finalize the model selection, especially in the case for convolutional neural network, which is so liberal and which have also like so many hyperparameters to fine-tune. And the beautiful part, or the part that I'm very proud of, is actually we also had an independent holdout test data, which means that we did not touch this data set until the second round peer review at media at that time, because we want to make sure the reviewers were okay with the model choice, which we used the training validation from the nested cross-validation. In this way, we can unbiasedly like estimate model performance. And we test like 3 different CNN architectures. The first one is we fit the entire 3D MRI into the 3D CNN. And the second architecture is what we call ROI, region of interest, or patch-based convolutional neural network. Because the motivation here is we know not all the brain regions which will be affected by Alzheimer's disease regarding brain atrophy patterns, and the most critical part might be the medial temporal lobe region and also the hippocampal region. That's why we might want to extract only the hippocampal or medial MDT, medial temporal lobe region, to fit into the neural network. And the third approach is the most common and like less computationally heavy method, which we first extract like those 2D slices from the 3D MRI and fit those slices into the 2D neural network. We answered several different questions. At that time, 2020, I think the deep learning, especially convolutional neural network for image analysis, has been becoming like very hot topic at the time. And the first question we answered is whether those deep learning models work better than conventional machine learning methods like linear SVM. Linear SVM has been introduced to voxel-wise, like high-dimensional imaging data classification for decades, and we know it's very robust regarding overfitting issues, etc. And from the data we had, like at that time, we showed actually even the most prominent convolutional neural network with the 3D ROI-based method, their accuracy, reported accuracy, is quite like similar to those simple linear SVM. And of course, the data sample, I think, is just like around like 2,000 subjects, which might be quite limited to maximize the power of deep neural network. The second question is like whether the model trained on one data set can be generalized to to other independent data sets. So here we train the model in the ADNI data set, which has a large sample size. And when we apply the 3D subject neural network to the ABLE and OASIS, we saw a big jump, big model jump for the OASIS data set. The underlying reason is actually we observe potential domain shift, and the domain shift is largely from the underlying like mismatch of the disease diagnosis, because OASIS, they don't have like mild cognitive impaired diagnosis. They put those MCI into patients like AD. And of course, if the ground-truth does not match with each other, of course, we saw the big jump of model performance. Another consideration is actually OASIS. The patients in OASIS data set is much older than those one from ADNI data sets. So finally, we showcased one specific scenario where data leakage can happen, like corresponding to the over-optimistic results observed in the table at the beginning. So in this figure, the upper level, the top figure here basically shows if we split the data, that's 2D slices from the 3D whole brain MRI, we first extract all the slices from all the people, and we split based on the slice, not on the people, on the person. That means the slices from one person's brain, they can go simultaneously to training validation. And we can observe that our orange for the validation, blue for the training, they both achieved very high accuracy. There is no overfitting issue observed at this stage before we apply the training model to the test. But if the lower part of the figure, if we extract the slices from all the people, but when we do the cross-validation splits, we split based on the people. That means the slices from the same people can only go to either training or validation. And by doing this, we can observe already the orange line for the validation. There's a big model difference, performance difference between the training and validation. We already observed like the overfitting issues here. But with the first approach on the top, you can only observe this overfitting issue model job when you apply the training model to the test. And that's potentially why, even in the table, a lot of those studies, first, they don't have an independent test. They don't really have a test data set. They only report validation as their test results. And of course, you don't observe the model overfitting with the validation data sets. This is a specific, like one specific scenario where data leakage can happen and provide over-optimistic results.
Okay, actually, quick question. What is the BA metric measure?
Sorry, it's balanced accuracy, which accounts for the imbalanced sample size between the healthy control and patients.
Okay, so it's accuracy weighted across the two populations.
Exactly.
Okay, and then yes, I'm seeing that the balanced accuracy on top is 0.76, except it's the actual value in the y-axis is higher. So how do we interpret that?
Can I repeat this question?
Well, I see the accuracy on the y-axis goes up, to 90 something, but you say BA is 0.76. So how do we interpret that?
This is from the independent. This is from the test data set.
Mm-hmm.
The BA number here, that's just one number. But the plot here I'm showing like during the APOCs training of the neural networks, the accuracy. I mean, yeah, I used accuracy here, but I should have already shown the accuracy, not balanced accuracy here.
Okay, so it's unbalanced. It's just for the diseased population.
No. What I mean, I think I used two different metrics here,
right,
for the training validation versus the test. But even you convert the balanced accuracy back to the accuracy, 0.76 is similar to this value for the accuracy values, I think.
Got it. Thank you.
Yeah. So then we made like all the code and results publicly available on the GitHub, and I'm very happy, still very proud for this study, like to have an impact on the field, because like I know like even right now people are using this framework like to apply those machine learning models to their own data. Then during my postdoc, I have been very interested in like extending like my machine learning skill sets to study specifically disease heterogeneity. This has been like showcased in the research of like Alzheimer's disease patients. We know like even under the same umbrella of the disease category like Alzheimer's disease, we observed quite big chunk of like heterogeneity regarding like brain atrophy pattern, regarding cognitive decline trajectories, regarding like online genetic factors. And here the motivation is to use machine learning models and brain MRI imaging data to derive the imaging-based disease subtypes, which I will introduce later. And during my postdoc in the team, we have both like we focus both on methodological development, and also like clinical application. We have developed a series of like machine learning based cluster optimizers, like the figure on the right shown. I mean, mathematically, this is just like clustering problem, and the most common approaches would be, for example, k-means, which is totally unsupervised learning, and you operate the algorithm like in the patient domain. But in that specific scenario, it might be less optimal because we know disease, any brain disorders, it evolves from different stage, like health control, mild cognitive impairment, then to AD on-site. And following this concept, what we propose like something different here, what we call the weakly supervised learning, our semi-supervised learning framework on the right side. Like unlike unsupervised learning, we are modeling here is the trajectory, what we call 1-to-k mapping from the reference group, normally the health control stage, to the patient domain. Assuming like we have k cluster or k subtypes, and this can be of course achieved by different machine learning approach, like generative modeling, like a GAN-based method, but also can be achieved like traditional deterministic like model, like SVM, linear SVM, for example. And then we apply those methods to different clinical population, try to understand like the clinical implication of those disease subtyping techniques. I will just show some example studies. The first one is a magic model. It represents basically multi-scale heterogeneity using like multiple linear SVM. Like I mentioned the figure in the previous like slide for the weakly supervised learning, the figure B here. Specifically, we are using like the linear SVM, which is perfect for this modeling scenario. We know like linear SVM provides a hyperplane to separate health control and patients, right? If the core part here is to construct a k-faced polytope to separate health control, CN, and the patients. And each hyperplane from each SVM, linear SVM, will create one face of this polytope. Let's assume there are two subtypes in the Alzheimer's disease. These two linear SVM creates one polytope, and we assign each patient to the nearest face of this polytope. In this way, we can do both simultaneously clustering and classification. And another novelty here is instead of using fixed number of brain features based on like brain atlas in the research field, instead we here use like what we call orthogonal projective non-negative matrix factorization, a variant of NMF, which has been shown to be data-driven and extract like part-based imaging feature instead of like PCA, which will just extract feature covering the entire brain driven by the variance in the data. And then we optimize the clustering solution at different scales from 32 to 1024. By doing this, we ensure like the finalized clustering solution is robust regarding the dynamic features of the dimensionality.
So wait, let me understand this. This looks complicated because first you are taking different parts of the brain, assigning them to a cluster, and then the clusterization of the whole brain is assigned to a cluster too?
No, the feature is extract at different scales, and you fit like each dimensionality of features from 32 brain, what we call structural networks, from 32 to 1024. And by optimizing each scale, we fix the rest of the scale. Like it's similar to those what we call coordinate gradient optimization. And then we ensure the joint optimization of the loss function between the different scale can converge at the end, like iteratively.
So then let me understand the relationship between the scales. Is it like hierarchical, where one unit at 32 becomes 2 units 64, or is it just 32, 64, no connection?
There is no concatenation of the feature set here. It's just we optimize, we ensure like the loss function or feature, the latent feature from the 32 can be leveraged by the next scale, like 64.
Okay, and you're trying to capture the spatiality of it. I mean, I guess it looks from your images, you have a little regions of the brain. Are you choosing them statically, or should I be thinking like a DB scan that figures out, this size of this region is homogeneous, this other one is homogeneous, and just builds out a spatial map?
That's a very good question. I mean, here we fix number like manually. We didn't, I mean, of course, like ideally you want to the model like get the optimal scales of the scale, like the choice of the number of features from each scales. But here we just basically fixed the number of features at each scale and iteratively optimizing like the loss function during the training.
Okay, thank you.
Then we apply these monsters to a clinical population of late life depression who had been diagnosed as major depressive disorder after 60 years old. And applying the brain MRI data, like the T1 weighted MRI, we found two disease subtypes or dimensions. The first one is dimension 1, which did not show any brain atrophy pattern. Instead, they show like a reserved brain volume in subcortical structure and bilateral like insular regions. And the second dimension, dimension 2, showed global brain atrophy that captured mainly cortical regions and some part of the insular regions. And after defining those neuroanatomical dimensions of the late life depression, we also looked at whether one of the dimensions showed microstructural change used diffusion MRI techniques. Like the figure on the right side of showing like only the dimension 2 showed large like widespread white matter disruptions over like in the white matter regions. And then we try to answer using the dimensional score of the two neuroanatomic subtypes as phenotypes. We link them to the genetics, perform genome-wide association, and we only found one putative like signal in the dimension 1 on the fourth like chromosome. And we look at the beta value, and it might potentially play a protective role against like the brain atrophy, which we do not observe like in dimension 1. But of course, remember the sample size here is quite like small because we restricted the population over 60 years old in the UK Biobank data. So this requires further validation when we have larger sample size. The second question is to apply the model to a longitudinal data from ADNI, because the motivation here is we know like late life depression and Alzheimer's disease has been mutual comorbidity with each other, and we try to understand first whether the two late life dimension progress like stably along 7 years follow-up using the ADNI data, like the figure on the right side, figure A, showing actually the expression of the two dimensions, especially for like 6 to 7 years old, are quite like stable, as we can see here. The same color bar following 6 to 7 years follow-up, they remain like quite stable, which means if one patient was assigned with dimension 1, then they are very likely to stay in dimension 1 with 6 years follow-ups. And the second question we are trying to answer, the figure B shows here, whether the dimension 1 or dimension 2 are more quickly going towards into AD diagnosis and also brain aging trajectory. The figure, as we expect, the dimension 2, which shows like at baseline, like widespread brain atrophy, is more quickly diagnosis converted like into AD diagnosis compared to the dimension 1. The same patterns has been observed with the SPARE BA score. It's a brain age metric derived from the brain MRI data too. So then after the SVM-based methods, we have also proposed like 3-GAN generalized advisory network-based approach following the same concept of the weekly supervised learning. This has been done by our previous PhD student at UPenn, like Yupen's lab, Zhijian Yang, who is right now as a research scientist at GE. But I think he might switch his job to Google, from what I understood like last time. Anyway, so we proposed like 3 GAN-based micers. The first one is from 2021, which we published at SLR. And this micer is called SmileGAN. It's exactly the same concept, but instead you constructing the particle 1-2K polytope. Here we just use the generative modeling the distribution from the healthy control to the patients. This is a perfect case for GAN-based modeling, and using only imaging data. And in a follow-up paper, we proposed a very similar micer using imaging MRI data only, called SerilGAN. Sorry, this paper published in SLR in 2023. So the concept here differs slightly compared to the SmileGAN. The SmileGAN is a very basic like clustering micers. We assume like each patient only express like one dominant imaging patterns. But here, SerilGAN is more like a representation learning micers, which assumes, okay, the same people, the same brain can express multiple dominated imaging atrophy patterns. This is slightly like modeling differences between the two micers, but they all use like brain MRI data. And then last year, we proposed a new method called the GeneScan, which is an extension from the SmileGAN method. Here, instead of modeling the 1-2K mapping using imaging data only in SmileGAN, in GeneScan, we introduce additional variance guidance by genetics, because we know like the imaging pattern has been substantially shaped by underlying genetics. That's the whole idea. We want to make sure that the derived subtypes is largely guided by imaging variance, but also like guided by genetics variance. This is specific case like for Alzheimer's disease research. So this is another study. We apply those GAN-based micers to a large aging population. And at UPenn, my previous mentor, he has been leading a large-scale like brain MRI consortium, which called iStaging. We have collected, like I think right now we have more than 50K people like who have at least T1 with the MRI scan. In this study, we basically include all those aging population, define what we call pre-aging, which younger than 50 years old and do not show any pathology, and also the aging population who are older than 50 years old, and they can have like MCI, they can have like Alzheimer's disease patients and any other pathology, and fit those data into our weekly supervised learning. The pre-aging as a reference domain, the aging population as a disease population, and we observed actually 5 different imaging pattern observed in this aging population. And for example, one result I want to highlight here is R2, the medial temporal lobe atrophy pattern, MTL atrophy pattern. And I just want to mention actually when we apply the method, we did not provide any prior knowledge regarding like Alzheimer's disease, but this pattern has been copes very well with like 80 hallmarks from the imaging atrophy patterns in the medial temporal lobe region and hippocampus region. And of course, when we perform the genetic association and biological pathway enrichment, we found this image pattern is highly enriched with dementia risks, amyloid and tau, and memory decline. I think this is a very interesting result showcasing that how Alzheimer's disease has been a very, very big driver in the aging population. So the third project.
So for this one, let me understand kind of the role of GAN, because previously you were focusing on explicitly clustering based, but it seems like you're using GAN not to generate images, but to cluster. Is that fair?
Can you re-articulate this?
It appears that you're using GANs for clustering, which is, I mean, normally GANs is you generate an image, that's, and you're trying to make fake images that look like real images. Yeah. But the application here is clustering, which is surprising.
The application can be clustering, like SmileGAN. It's a hardcore like hard-coded, like subtype 1, subtype 2, or cluster 1, cluster 2, cluster 3. But the serial GAN is slightly different. Like the assumption is, okay, the same people, they can have 55% of R1 dimension, 40% of R2 dimension, etc., etc. So it's more like representation learning. But you are right, GAN is known like for distribution generation. Like, okay, it's the same idea here. We use the real health control data to simulate the synthetic patient data, and the discriminator to make sure the simulated patient, the studio patient data is similar to the real patient data. It still follows the concept of GAN.
Okay, so yeah, so I should be thinking of, you have brain images from healthy people. The GAN generates the same healthy images. Discriminator says which one is which.
Yeah.
Then what? Because now you've gotten a good generator. Well, actually, is this to make a good generator, or is this to make a good discriminator? Like, what are you using from the architecture?
So the clustering part is nothing related to that. I mean, the clustering part is actually after you generate those pseudo patients in the patient domain, we have a inverse mapping in the method called Z, like Z, denoted as Z parameter. We inverse map this to the clustering domain, and that's where the clustering is performed. It's not like the clustering is not like inside the generator. So after the discriminator, we have the latent space. We perform the inverse mapping into a clustering domain, and that's where like the clustering has been assigned to different patients.
Okay, so I think I'm following. So you're taking in the discriminator, it has to create a latent representation of any image it gets. Yeah. You're clustering the latent representation of the discriminator.
Yeah, the latent place. Yeah, exactly. The latent space generated by the discriminator, we mapped back to another the inverse space, and that's where the clustering is performed.
Cool. So then, yeah, this is like this standard like super PCA essentially. But then why not PCA or autoencoder or many other ways of getting a latent space?
Exactly. I mean, PCA is just linear combination, right?
Yeah, but like autoencoder would be the standard way of doing it. GAN is more complicated. So why GAN, not autoencoder?
We actually use like we tried like autoencoder, even like, but actually we compared the results for autoencoders. I remember Zhijian at that time, he told me the method is not as stable as GAN, relatively speaking.
Interesting. Okay, cool. And so, and the applications primarily are these, you want to reduce the dimensionality of disease progression for downstream diagnostic applications, not so much for generating images or anything of that nature.
Exactly. And in the paper we show, I mean, I didn't show here, but in the figure, we basically showed using those dimensional score R1, R2, or the P1 for the cluster, you can predict disease progression. You can predict amyloid load and tau load, like from health control to the patients.
Got it. And actually, since we're on this, because you can generate images, is this a way of generate synthetic data to like rebalance your data sets? Because you do have this problem, far fewer diseased patients than you have healthy patients. But if you can synthetically generate data, you can actually rebalance your data. For sure.
Yeah, definitely. But like I mentioned, like the generating of the pseudo patient data is not the main objective here. And another thing I forgot to mention, actually, the imaging feature is not the raw imaging feature here. Is extract the brain volumes, like tabular data. It's not the voxel-wise imaging. Of course, we can use the voxel-wise imaging, but the mode clouds, all those things, is going to be more like prominent in this high-dimensional data, I think.
All right, thank you.
So then I like at the end of my postdoc or towards my independence, I have been like, I want to do something new beyond like all the previous research. That's where I some like very like landmark paper in the field draws my attention. The two papers I want to highlight a little bit is for the brain imaging genetics research. Like all my previous research has been focused on brain MRI, and at that time I think with the availability of the UK Biobank data, specifically from the first paper I think from Nature 2018 from Smith and Elliott colleague from Oxford, they have linked those large scale, because in UK Biobank we have more than 50K brain MRI, and they have derived what we call imaging derived phenotypes, IDP, and using like T1 weight MRI, resting state function MRI, diffusion MRI for DTI or multi-shell diffusion matrix, etc. And they link those more than 3,000 IDP with those underlying common genetic variants using like genome sequencing technique provided by the UK Biobank data. And they showed actually largely the imaging features has been largely like decided by the underlying genetic architecture of those imaging features. And another paper from 2024 from Junhao who is right now at UPenn, we collaborated some of our previous work on brain aging, and he has been leverage UK Biobank imaging genetic data to establish cross-organ interconnection. For example, the paper, the brain-heart connection, published in Science. And this gave me a lot of like inspiration for my future research direction. And of course, they have already done like fabulous work on this research direction. And at that time, I want to do something like more slightly different or more novel. In my personal opinion, that's where I want to bring in like machine learning topic in this research brain imaging genetic field, using AI and machine learning to define some data-driven machine learning biomarkers based on imaging or proteomics, for example, and then link them to underlying genetics to discover underlying biology of aging and disease. So the first paper I did at that time was an extension from what I call the magic model, where we derive those using like a specific variant of NFM misers, what we call stochastic non-negative matrix factorization. Basically, we trained the model as a deep learning model in specific case because the sample size here is more than 50,000 K. If we apply those standard NMF misers, it's a batch learning miser, so the memory issue is a big challenge. So at that time, we made some improvement for the OPNF miser by fitting small batches into the training misers like a deep learning iteratively, and that's where we overcome the computing challenge regarding the memory. And then we extract those what we call patterns of structure covariance networks at different scales. Then downstream association analysis is to link those NMF-based structural networks with common genetic variants. We hypothesize actually compared to the the brain atlas-based approach, those imaging features used in the two papers previously mentioned, those data-driven imaging features might provide higher statistical power to discover new genomic signals. And this is largely reflected by our study showing that actually a higher number of genomic loci is identified by the NMF imaging features. And I mean, I don't know you guys if you guys are familiar with the GWAS. Basically, genome-wide association is just an association, either by linear regression or logistic regression, depending on your phenotypes, continuous variable or categorical variables. And with such a large sample size in the UK Biobank, more than nearly like 50,000 people, of course, you can find some statistically significant signals, the genome-wide significance, but that does not imply any like biological interpretation or especially causal implications. So there are several different post-GWAS analyses try to partially validate those genetic signals. Here is one example, what we call biological pathway enrichment analysis. So let's take the feature, the example on the right side. So this is one of the components from the 128 scales, and it basically captures the medial temporal lobe region and hippocampal region. I like this example because everybody like in the AD research field knows this is a very robust hallmark of Alzheimer's disease. And we first perform the GWAS and then map those SNP signals back to the gene, which gene has been like linked to this imaging pattern. And then we use those genes perform biological pathway enrichment analysis. And we largely found, okay, this gene has been enriched, for example, Alzheimer's disease, which totally makes sense because This is a hallmark of RSM disease. And also, of course, this has been implicated in other biological pathways like nuclear membrane, etc. So this is just one specific example of how we can leverage different post-GWAS analysis to validate those results. I will also showcase some other post-GWAS analysis in the following slides with other projects. So this is a paper we published like 2 years last year, early like in March last year. So we extended like the concept of imaging genetics. Here we're using first brain MRI, like green matter from T1 weighted MRI, white matter from diffusion MRI, like DTI techniques, and the functional connectivity from the resting stage function MRI from the UK biobank data. We derived those 3 brain age, what we call multimodal brain age, and then we do the same thing to link those brain multimodal brain age gap with common genetic variants, perform like biological pathway analysis, etc. And again, this is like a more like aligned more like my current research, like focusing on human aging and disease beyond the brain. And the overall motivation has been the fact that we know like RSM disease or even other like, for example, systematic disease like diabetes, they have been implicated like in different organ dysfunctions. And with the current like availability of the multi-organ, multi-omics data, like the big data initiative like UK Biobank, we have a good opportunity like to model human aging and disease like more holistically and comprehensively. In a follow-up study, we extend the brain age gap projects to the whole body system, including in total like 9 organ system: brain, eye, immune, cardiovascular for the heart, musculoskeletal, for example. And we derive those biological aging clocks, what we call phenotype-based aging clocks, because many of those organ age is derived from some lab results. For example, the musculoskeletal is from like your body weight, your height, etc. And then we perform a large-scale GWAS analysis. This is another case to partially validate those genetic signals, what we call Mendelian randomization, which basically use the SNP as an instrument variable to mimic like randomized clinical trial, try to understand potential causal relationship from the different non-organ specific agent to some other clinical traits like disease endpoints, like RSM disease, and also lifestyle factor like sleep, like body weight, etc. And as we can show, one specific example I want to highlight here is RSM disease has been shown with our Mendelian randomization results being positively associated with brain age gap. The brain age gap is also negatively associated with sleep duration, like how many hours you sleep like during the night. Another example, I think, which is very important, is the body weight. As we can show, body weight has been implicated or causally linked to different organ systems like immune, pulmonary, metabolic, etc. This just showcases like how potentially like better management of your lifestyle factors could like improve your overall like organ aging health.
So how did you establish the causal links? Because these are, these are large data sets, but not that large, especially given how many dimensions you're talking about. And they tend to be sparse because I believe these are patient records. So many factors are not documented.
So that's a good part of genetic analysis. The specific methods, like I mentioned, is called like Mendelian randomization. You don't need the individual data. You just need the GWAS summary statistics. And basically it's testing the SNP, let's say RSM disease and the brain age. You perform GWAS for RSM disease, you perform GWAS for brain age gap. Then you look at those beta value between the SNP versus the trait. That's going to be the case, SNP versus AD, SNP versus brain age. And they basically test the association of the beta values between the 2 GWAS. Does that make sense?
So let me just answer the association, meaning that so beta values is a significant score, right?
No, the beta value is the effect size from your linear regression or longitudinal regression.
Okay, good. So this is the effect size, and this is still before multiple hypothesis testing. So just the raw effect size, right?
Yeah, exactly.
Cool.
I mean, yeah.
And now you're trying to say that for which genes, these are high effect size for both of these phenotypes.
We don't look at specific genes. So basically here is testing, let's say after merging the two GWAS, the population, we define like 20 SNPs. The 20 SNPs has been shown like association with AD and also brain age gap. So here I just plot the beta value in a scatter plot showing the beta value of a scatter plot of the beta values for the two phenotypes. Then you see how the two set of beta values are linearly correlated. That's basically is testing the relationship of the beta values
Across different SNPs.
I might have a figure.
Oh, that'd be great, yeah, because it also sounds like you're going to have one dimension for every one of these phenotypes, right?
Let me show you here.
Oh, you may have to show your whole screen because I think you're only sharing your presentation window.
Okay, let me find first the plot I want to show. Okay, how should I reshare the screen?
I'll just share the whole screen. That'll just work, because then we'll see all the windows.
Okay, let me stop sharing first. Can you see my screen?
Yes.
Right? So the figure A here is testing the SNP effect on AD. That's the AD GWAS. We pick up each dot here is a SNP. In this case, I don't know how many SNPs. Let's say 20 SNPs. So those 20 SNPs, they have been significantly associated with AD on x-axis. On y-axis, let's say this is a digestive aging clocks. Those SNP has also showing like a significant association. So what Mendelian randomization is testing whether the two set of beta values they are positively associated or negative. In this case, we show like positive association, but of course we observe like one outlier here. That's a concept of Mendelian randomization.
When you say outliers, so I mean, should we be seeing that these are all strongly, and then when you say outlier, what's the dimension? What do the two dimensions show? Like it's intuitively it would be there's strongly associated with both of them, right?
Yeah, I mean here is what I mean outlier. It's just like these specific SNPs on the right upper corner here is far away from the rest of SNPs. This might be true by biological signals, but in genetics, it's very unlikely. That's what I mean. I mean, like.
Okay. And what are the two dimensions measure? Is it the beta?
Yeah, the AD, the GWAS.
Oh, the extent of the impact on AD. Okay.
Yeah, the AD GWAS. So you basically linked the disease diagnosis with common genetic SNPs. You do the same genome-wide association with your aging clocks on the y-axis.
Okay. And then, so what's the significance now that you have a few genetic SNPs that are related to both? Is it like analysis, further study analysis, like or does it indicate something about the mechanism?
This indicate potential causal relationship.
Okay, potential
causal relationship, but I mean, Mendelian randomization, they have very strong statistical assumptions. Like the outlier, for example, here, they might violate the statistical assumptions. This is population genetics, like statistical method, I think. I do.
Got it. And you can't do any causal interventions here.
This has some implication, for example, for whether we can like for those genetic hits, the SNP, the genes, if we found this is very strong like causal signals, causal evidence here. We can actually use those specific location gene for drug repurposing, for example, for white lab iPSC mouse model to validate like those signals.
Got it. Okay, thank you.
Okay, let me share back my screen. Yeah, this is another result which I like a lot. It's just to test like an existing hypothesis, what we call, oh, what happened?
Oh, no, we were able to see it.
Can you see it? Okay, sorry. Yeah. So basically, this is a very well known like conjecture. It's called like Chevrolet conjecture, which was from like a French scientist like 2 years ago. He is like a geneticist. So in this specific case, we derived those organ aging clocks from the non-organs, and basically he suggests, well, if we only have the genetic data between two clinical traits, like let's say the heart, the eye organ aging and the brain organ aging, we only have the population genetic data, but we don't have the phenotypic data. To look at the phenotypic correlation, you can perform like Pearson correlation R, very simple, right? You perform Pearson correlation R, they give you like a coefficient. But he suggests if we have the online genetic data, we can actually use those genetic correlation, what we call genetic correlation, to look at to use this as a proxy for their phenotypic correlation. Like the figure on the upper triangle part is basically shows the phenotypic correlation for the non-organ aging clocks, and on the lower triangle part shows the genetic correlation. Let's see, for example, for the eye, we can see between the eye and the brain. The brain and the eye show both positive phenotypic correlation and genetic correlation. This kind of like to support like his hypothesis like 200 years ago, which I like a lot. And of course, one might ask whether there's some exception for this kind of results. My answer is yes, but I didn't show it here. But with another paper, we used like disease subtyping for different like brain disorder, like autism, spectral disorder, schizophrenia. We definitely saw like opposite direction between the phenotypic correlation and genetic correlation. And the reason why we observed those opposite directions in those estimation is largely influenced by the environmental factors, which totally makes sense in psychiatric disorder, because we know, like, of course, we cannot say like autism is totally decided by your genetics, right? Because like environmental factor, lifestyle factors also plays a role. That's where I think if strong environmental factors influence someone's like overall like health, this can like inverse like the directionality of the phenotypic correlation. So I will also briefly introduce some other research we have done in the past year, looking at what we call biological aging clocks. This is an extension from the previous like multi-model brain age, and also the phenotype-based brain aging clocks, biological aging clocks. Sorry. So here we extend this concept to plasma proteomics data, which is quite a hot topic in the field, I think. So I don't know if you guys are familiar with the biological age clock concept. Basically, it's very simple. We can use any like biomedical data, like brain MRI or plasma proteomics, metabolomics, using those as features in a regression model, machine learning model, by I don't know linear regression, etc., and to predict your chronological age. And of course, your model is not gonna perform like perfectly. There will be some mismatch, and what we call the AI predict methods, predict age minus your true calendar or chronological age. That gap, that difference, we call it like biological age gap, BAG. And in this specific case, in this paper, I highlight several different aspects regarding methodological and clinical consideration to derive and using those like biological age gaps. And one specific technique here, what we call is age gap, age bias correction. What is age bias? Basically, when you perform a linear regression from a statistical perspective, is you always like underestimate your predict age, like on the first column, the up first column figure here. In your model, that's basically the best scenario your model performs always happen in the middle of your data. Let's see, the population here is from 40 years old to 70, and the best model is always around like the 45 years old. That's like regression towards the mean. And in the younger population, you underestimate your age, and in the older population, you always like overestimate your age. That's what we call age bias. And in the brain age community, we have proposed like different like measures to correct for this residue. The issues here on the top figures here on the right side, if we don't perform age bias correction, normally we expect those age gap should be like lower in healthy population and should be higher in the disease population, like Alzheimer's disease patient population. But if like I shown here on the upper part of the figure, if we don't perform age bias correction, we show the opposite trend, which is quite like counterintuitive. Basically showing like, for example, Alzheimer's disease patients, they show lower biological age gap, which looks younger, which does not make sense, right? And on the second row of the figure, the right side, after correcting the age bias, we can show the more plausible trend showing like higher BAG, biological age gap in the diseased population. This is just one specific scenario when, I mean, machine learning right now has been widely used in any biomedical research. And I just want to make sure people know what they are doing and how to correctly like develop those aging clocks and interpret them.
So this sounds a lot like, well, causal inference or like a treatment effect estimation, where you have to like just separate, for example, separate your different populations, the disease, the healthy or condition and progression of disease. Is that the kind of approach we should be thinking of?
Yeah, exactly. And also I forgot to mention, actually, the model is trained only in the healthy population. And then when you apply those age clock model to disease population, of course, you'll see some difference, right? And we believe those differences might be driven by the underlying pathology.
Okay, that makes sense. Yeah.
And this is another example, like we look at the influence of different elements when you develop such like aging clocks, for example, sample size. We observed overfitting issues regarding how many proteins you fit into your model. The more proteins you fit in model, of course, you are more likely to see the overfitting issues because a lot of those proteins, they are highly correlated. And for example, whether to combat this overfitting issue, whether you reduce the number of features, that's what we call organ specificity. Because those plasma proteomics circulate in the blood, in the whole body, and we know some proteins, they are overexpressed in specific organs. And by picking up those organ-specific like proteins, we can train organ-specific aging clocks. And this also helps like prevent like overfitting issues. And third element, for example, a lot of folks in the biological aging research field always argue, okay, your machine learning model do not perform very well predicting your chronological age. One typical like metric, what we call mean absolute error, is roughly like in UK Biobank is like 5 years old. That means at the population level, your model, the errors is around like 5 years old, younger or older than your chronological age at the population level again. But a lot of reviewers argue, okay, why don't you use like deep learning methods to achieve like lower MAE, like 2 years, 1 year? That they argue that might be better. And here I showed basically that's not the main objective of developing such aging clocks model. And for example, the figure D here, like as this one. So the figure D here, basically I tested like the aging clocks derived at different epochs from figure C. You can see the loss function with more epochs. In general, it's get better, like lower MAE. But if you link those aging clocks with cognitive score, one cognitive score, like in the figure D here showing like the standardized beta value, the sample, the effect size, we can see a typical like U-shaped pattern, which means at a certain point, the benefits of obtaining a better model, meaning like lower MAE, saturated around like 500 epochs. If you train furthermore, like the model achieving lower MAE, lower loss function, lower loss, you can see the benefits like decreased. That's one specific scenario showing like the objective of developing such age prediction model might not be to predict your calendar urge per se. Instead, we should focus on the predictive power for other clinical variables like mortality, like cognitive decline. So in an earlier paper we published like several months ago at Nature Medicine, we extend this concept to different organ imaging beyond like brain MRI. In this case, we have like abdominal MRI from liver, kidney, spleen, and also heart MRI, etc. We're not going into details because of the time, but very similar pipeline from our previous aging clocks. To further enrich the biological aging clocks from the team we developed, here we basically extend the same concept to plasma metabolomics data, like the small metabolites. And of course, again, this plasma metabolomics data circulate the entire brain, and selectively we can pick up what we call organ-enriched metabolites. But of course, in this scenario, because we know those metabolome data from the plasma largely target like the cardiometabolic system. So here, for example, we only focus on the immune hepatic, like digestive, metabolic, and endocrine systems, like not for the brain, for example, for the eye, for example. In another paper we published earlier in Nature Biomedical Engineering, it's kind of looking at the raw imaging feature level, but here instead of using the brain atlas-based features, we use those NMF-derived structural network features linking like 3 organs, like the brain, the heart, and eye, using like imaging data, genetics data, and proteomics data to construct a brain-heart-eye axis. And we largely showcased actually at the imaging level, at proteomics level, those cross-organ connections. And one important message here, which I did not show, but in the original paper, we showed actually combining those imaging features from different organs, but also from different omics, like genetics, imaging, and proteomics, boost like prediction power for disease, for cognitive decline, and for mortality. And that highlights basically the one potential benefit of cross-organ and multi-organ modeling in human age and disease. Finally, to end this presentation, I just want to highlight I'm also like a big fan of the like data sharing and public like reproducible science, open science. So to disseminate the results and the GWAS summary statistic masters and the pre-trained AI masters, we have developed like several different public portals. And I'm very happy like to say like researchers from the field, especially those geneticists, which have been like downloading the GWAS summary statistic for their own research purpose. Finally, I want to thank you all my collaborators from Penn, from NIA, from University of Melbourne, and USC, and ICL. I would be very happy like to answer any question if you have any. Thank you.
Well, yeah, thank you very much. It was fantastic. I think we're already past time, so I think we'll wrap up here, but very like diverse set of topics. Thank you.
Thank you.